Hi everyone, welcome to my YouTube channel @NEET Aspirant #biology. Hope all are doing well. So our today's chapter is biological classification, that is chapter number 2 of biology class 11th. Chapter number 1, the living world, is already uploaded on my channel. If you have not watched that video, then please watch. Before start the video, I requested to all, please subscribe my channel and share the video with your friends, and also comment below the video. Now we will start. So our today's chapter is biological classification. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific, but born out of a need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. In Linnaeus' time, a two-kingdom system of classification with Plantae and Animalia kingdoms was developed that included all plants and animals respectively. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic green algae and non-photosynthetic fungi organisms. Classification of organisms into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two-kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationships, etc. Classification system for the living organisms have hence undergone several changes over the time. Through plants and the animal kingdom have been a constant under all different systems. The understanding of what group organisms be included under these kingdoms have been changing. The number and nature of other kingdoms have also been understood differently by different scientists over the time. R.H. Whitaker, 1969, proposed a five-kingdom classification. The kingdoms defined by him were named Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for classification used by him included cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationships. Table 2.1 gives a comparative account of different characteristics of the five kingdoms. The three-domain system has also been proposed that divides the kingdom Monera into two domains, leaving the remaining eukaryotic kingdoms in the third domain, and thereby a six-kingdom classification. You will learn about this system in detail at higher classes. Let us look at this five-kingdom classification to understand the issues and considerations that influenced the classification system. Earlier classification systems included bacteria, blue-green alga, fungi, mosses, ferns, gymnosperms, and the angiosperms under plants. The character that unified this whole kingdom was that all the organisms included had a cell wall in their cells. This placed together groups which widely different in other characteristics. It brought together the prokaryotic bacteria and the blue-green alga, Cyanobacteria, with other groups which were eukaryotic. It also grouped together the unicellular organisms and the multicellular ones. Say for example, Chlamydomonas and Spirogyra were placed together under alga. The classification did not differentiate between the heterotrophic group fungi and the autotrophic green plants. Through they also showed a characteristic difference in their wall composition. The fungi has chitin in their walls, while the green plants had a cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom, kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista has brought together Chlamydomonas, cholera, either placed in alga with plants and both having cell walls, with paramecium and amoeba, which were earlier placed in the animal kingdom which lack cell wall. It has put together organisms which in earlier classification were placed in different kingdoms. This happened because of the criteria for classification changed. This kind of change will take place in future too, depending on the improvement in our understanding of characteristics and evolutionary relationships. Over time, an attempt has been made to evolve as a classification system which reflects not only the morphological, psychological, and reproductive similarities but is also phylogenetic, that is based on evolutionary relationships. In this chapter, we will study characteristics of kingdom Monera, Protista, and fungi of the Whittaker system of classification. The kingdom Plantae and Animalia, commonly referred to as plant and animal kingdoms respectively, will be dealt separately in chapter 3 and 4. 2.1 Kingdom Monera. Bacteria are the sole members of the kingdom Monera. They are the most abundant microorganisms. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot springs, deserts, snow, and deep oceans where very few other life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under 4 categories based on their shape: the spherical Coccus, the rod-shaped Bacillus, the comma-shaped Vibrio, and the spiral Spirillum. Through the bacterial structure is very simple. They are very complex in behavior. Compared to many other organisms, bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substrates. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs, that is, they depend on other organisms or on dead organic matter for food. 2.1.1 Archaebacteria. These bacteria are special since they live in some of the most harsh habitats such as extreme salty areas, halophiles, hot springs, thermoacidophiles, and marshy areas, methogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant animals such as cow and buffaloes, and they are responsible for the production of methane biogas from the dung of these animals. 2.1.2 Eubacteria. There are thousands of different eubacteria or true bacteria. They are characterized by the presence of a rigid cell wall and, if motile, a flagellum. The cyanobacteria, also referred to as blue-green alga, have chlorophyll similar to green plants and are photosynthetic autotrophs. Figure 2.2. The cyanobacteria are unicellular, colonial or filamentous, freshwater, marine or terrestrial alga. The colonies are generally surrounded by gelatinous sheet. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocytes. For example, nostoc and anabinia. Chemosynthetic autotrophic bacteria oxides various inorganic substances such as nitrites, and ammonia and use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legume roots, etc. Some are pathogens causing damages to human beings, crops, farm animals, and pets. Cholera, typhoid, tetanus, citrus canker are well-known diseases caused by different bacteria. Bacteria reproduce mainly by fission. Figure 2.3. Sometimes under favorable conditions they produce spores. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to the other. The Mycoplasma are organisms that completely lack a cell wall. They are the smallest living cells known and can survive without oxygen. Many Mycoplasma are pathogenic in animal and plants. 2.2 Kingdom Protista. All single-celled eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. What may be as photosynthetic protistans? To one biologist, may be a plant to another. In this book, we include Chrysophytes, Dinoflagellates, Euglenoids, slime molds, and protozoan under Protista. Members of Protista are primarily aquatic. This kingdom forms a link with the other dealing with plants, animals, and fungi. Being eukaryotes, the Protestant cell body contain a well-defined nucleus and other membrane-bound organelles. Some have flagella or cilia. Protists reproduce asexually and sexually by a process involving cell fusion and zygote formation. 2.2.1 Chrysophytes. This group includes diatoms and golden alga desmites. They are found in freshwater as well as in marine environments. They are microscopic and float passively in water currents, plankton. Most of them are photosynthetic. In diatoms, the cell wall form two thin overlapping shells which fit together as in a soap box. The walls are embedded with silica and thus the walls are indestructible. Thus diatoms have left behind large amount of cell wall deposit in their habitat. This accumulation over billions of years is referred to as diatomaceous earth. Being gritty, this soil is used in polishing, filtration of oil and syrup. Diatoms are the chief producers in the ocean. 2.2.2 Dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue or red depending on the main pigment present in their cell. The cell wall has stiff cellulose plates on the outer surface. Most of them have two flagella. One lies longitudinally and the other transversely in a furrow between the wall plates. Very often red dinoflagellates, for example, goniolics, undergo such rapid multiplication that they make the sea appear red. Red tides. Toxins released by such large numbers may even kill other marine animals such as fishes. 2.2.3 Euglenoids. Majority of them are freshwater organisms found in stagnant water. Instead of a cell wall, they have a protein-rich layer called a pellicle, which makes their body flexible. They have two flagella, a short and a long one. Though they are photosynthetic in the presence of sunlight, when deprived of sunlight, they behave like heterotrophs by predating on other smaller organisms. Interestingly, the pigments of euglenoids are identical to those present in higher plants, for example, Euglena. 2.2.4 Slime molds. Slime molds are saprophyte protists. The body moves along decaying twigs and leaves iglifying organic material. Under suitable conditions, they form an aggregation called a plasmodium, which may grow and spread over several feet. During unfavorable conditions, the plasmodium differentiates and forms fruiting bodies bearing spores at their tips. The spores processes through walls. They are extremely resistant and survival for many years even under adverse conditions. The spores are disappeared by air currents. 2.2.5 Protozoans. All protozoans are heterotrophs and live as predators or parasites. They are believed to be primitive relatives of animals. There are four major groups of protozoans. Amoeboid protozoans. These organisms live in freshwater, seawater or moist soil. They move and capture their prey by putting out pseudopodia, false feet as in amoeba. Marine forms have silica shells on their surface. Some of them, such as antiamoeba, are parasites. Flagellated protozoans. The members of this group are either free-living or parasitic. They have flagella. The parasitic forms cause disease such as sleeping sickness, for example, trypanosoma. Ciliated protozoans. These are aquatic, actively moving organisms because of the presence of thousands of cilia. They have a cavity gullet that opens to the outside of the cell surface. The coordinated movement of rows of cilia cause the water laden with food to be stirred into the gullet. For example, paramecium. Figure 2.4d. Sporozoans. This include diverse organisms that have an infectious spore-like stage in their life cycle. The most notorious is Plasmodium, malaria parasite, which causes malaria, a disease which has a staggering effect on human population. I hope everyone enjoyed the video. Tomorrow we will start from 2.3 Kingdom Fungi. Please share this video with your friends and like